There's actually several different definitions of proteins. The most pertinent definition of a protein is it's a molecule in our body that does stuff. Those physiological functions are always involving some kind of protein or some set of proteins. There are structural proteins such as collagen. You may have actually heard of that before. The function would be something like mechanical support. There's enzymatic proteins. So these would help to catalyze reactions, either breaking structures apart perhaps, like a disaccharide being broken into monosaccharides. There could be transport proteins. This is probably the best known protein in your body, which is hemoglobin. Hemoglobin is the oxygen carrier. It can actually carry 4 molecules of oxygen. And so this entire structure here is called hemoglobin. I want to point this out. The hemoglobin ends with the letters IN. In this class, if you see a word ending in IN, that is a protein. I haven't ever come across a word that ends in IN that is not a protein. So just keep that in mind. So transport proteins will move things around, such as moving oxygen through your blood, hemoglobin. The next type is a contractile protein, myosin, actin. These are 2 proteins that are responsible for muscle contractions, along with a couple of other ones that we will be covering. Communication proteins, super important. Go right to the word receptor. A receptor, it's actually right in the word. The receptor receives information. So it receives, for example, this might be a neurotransmitter that is attaching to this receptor and sending a signal into the cell that something has to happen. So the receptor would be the protein. And then, these are important right now in the world, antibodies. Antibodies, these little Y-shaped guys, all these little blue guys, these tiny little structures are actually free-floating proteins, usually free-floating, not always, but usually free-floating. You search out and hunt down viruses and bacteria and other pathogens that might attack your body. So antibodies are super important. So the little Y-shaped guys here, these are also proteins. So this gives you an example of the ways that proteins do stuff in your body. Now, another way to describe proteins would be to describe them by their monomer versus polymer structure. Here we have a familiar situation. We're actually just showing both of them at once. We have a dehydration synthesis where we take 2 monomers of protein and put them together. Now the monomers are called amino acids. There's 20 amino acids with some derivations there, but the main amino acids, there's 20 of them. And I'm not going to make you memorize all the amino acids, but I actually would like you to be able to draw this. I would like you to see the general shape of an amino acid. This is the amine end, NH2. We have a central C with an H. And then we have a carboxyl group on this end, which is a C, double O, single, double bond O, single bond OH. That is called a carboxyl end. And so there's an amine end, and there's an acid end. And the way that it works is, and then there's an R group. The R group is a variable group that has a different quality, that some of them have a charge to them, some of them are polar, some of them are non-polar, some of them have special qualities such as a sulfur. If there's a sulfur in there, then that does special stuff too, which I'll talk about in a minute. Okay, so we have these 2 amino acids. They are able to bond together. There's the H. There's the OH. The OH of the carboxyl group is attracted to one of the H's of the amine group, and this is how they actually bond together. Look at that. We removed the water. This is a dehydration synthesis. They've synthesized together to what we call a dipeptide, because this guy right here is called a peptide bond. Peptide bond. So what will happen is you'll end up with a string of amino acids, one attached to the next, attached to the next. The amine end is attracted to the carboxyl end, so that peptide bond always happens the same way. If you have many amino acids in a row, the carboxyl end is attracted to the amine end with this peptide bond. So in this picture, we have 5 amino acids and 1, 2, 3, 4 peptide bonds. You'll notice that the R groups actually aren't involved in the peptide bond at all. They simply hang off of that central carbon and allow the amino acid to have whatever special quality it has, whether it's charged, whether it's polar, whether it's nonpolar. And that's what starts to differentiate these guys from each other, is that R group. Long chains of amino acids connected by peptide bonds. All right, so the last way that we would define a protein, and there's a few more definitions too, but one of them will come down to the central dogma of biology. If you want to write in your notebook DNA at the top, skip a line down, write RNA right underneath of it, skip a line down, write amino acids. So the DNA, you could put in parentheses a gene, G-E-N-E. Draw an arrow down to that RNA, and next to the arrow write transcription. From the RNA, draw an arrow down to the amino acids and write translation. So DNA, RNA, amino acid sequence is the way that proteins are actually created. That amino acid sequence is entirely dependent on the DNA properly encoding what the eventual protein is supposed to look like. And given the correct temperature and the correct pH, it will always fold the same way. So if we were to define proteins this way, we would probably say that proteins are the end product of a gene. You end up with this, what we call the primary structure of a protein, which is simply the string of amino acids. The secondary structure, there's really only two ways you can have a secondary structure. The primary chain starts to kind of fold in on itself, mostly due to the hydrogen bonds. So we mentioned this in a previous video. I'm going to point them out now. So when we take a look here, the oxygens are denoted in red, the hydrogens are denoted in blue. You'll notice that there's a little dotted line between that red and that blue. You may recall that oxygen carries a slightly negative charge. The hydrogens carry a slightly positive charge. And so they have this just slight bond between them. In secondary structure, you can have an alpha helix. And again, take a look. The amino acids are actually, the hydrogen bonds are actually really predictable. And then the same here. This is called a beta sheet. And the beta sheet also has, look at this, little hydrogen bonds that kind of force this configuration. So this would be the secondary structure, either an alpha helix or a beta sheet. Now the tertiary structure, now it starts to actually look like a protein, where the alpha helices and or the beta sheets are folded up to form a compact globular molecule that's held together by intramolecular bonds, intra meaning inside. So inside of the molecule you have other bonds as well. Now, inside of here you'll see that there are alpha helices, there's some beta sheets in there, but then these start to fold in on themselves in kind of unique ways. For example, if you had R groups that are polar, they would tend to be attracted to other R groups that are also polar, or if they were nonpolar, they would be attracted to other R groups that are nonpolar. If you had, for example, a special amino acid such as one that contains a sulfur, remember I said that sulfur is an important part of proteins. Now that would be called a sulfide group. Sulfurs tend to be attracted to other sulfurs. And so what that does is as the R groups are attracted to other R groups, they tend to really force a particular shape to take place in the eventual protein. The shapes of proteins are very, very important. Actually, the shape of a protein describes its function. So for example, this guy right here, one of the things that sticks out to me is there's kind of like this little divot. This might be, for example, a receptor. There's something else that's supposed to fit perfectly into that shape. And if anything is a little bit off, where the amino acids are not bonding correctly, possibly due to a mutation all the way back at the level of the gene, then the shape of the protein might be off and it might no longer have the functionality that it had previously. And then lastly, this last one that we're looking at is quaternary structure. Now this by itself, it would be one protein. But if you have this guy interacting with another protein, these would then be called subunits, or they might be called polypeptides. And then they were combining together to make one protein. So here we have a massive protein that's actually made of two subunits, each of which are kind of their own protein, but together they have a different functionality.